we're going to go pretty heavy now into the bioassay biomarker portion of this. Now, talk to our audience, especially about the biggest challenges in early therapy development as it relates to the topic of bioassays. So where are we with that?
Sure, particularly in cellular therapy. The biggest assay that we talk about is generally the PK assay, which in cellular therapy is referred to as the vector copy number. It's how many CAR T cells did you put into the patient, and then how many are there in each point in time longitudinally. And like one of the Q&A questions says, the good news is that's generally a analytical development release assay, measuring the CAR T cells. And so we in translational can leverage a lot of the foundational work they do and then deploy that on the blood samples that we collect in the course of our clinical studies. Now, because we're a small, nimble biotech company, we don't generally trip over the bureaucracy and the silos that you might find in big pharma companies. But the advice that I could give, having worked in big pharma in the past, is having one version of the truth, one assay, is always the right way to go. And the question just becomes, where does that assay live? Does it live in the preclinical or manufacturing analytical release space? Does it live in the translational lab, or does it live at a third-party outsourced vendor? But if you can get all three groups to agree that this is the right assay, we've all validated independently, we've all generated a bunch of paperwork on it, then at that level, you'd have to argue it doesn't matter where it resides. You send all your samples there to be assayed.
Gotcha. Okay. Doug, I want to get your perspective now from a gene therapy point of view. Same topic here around bioassay and kind of the biggest challenges in early therapy development. What are you seeing on your end?
Yeah, I would say specifically related to bioassay development, one of the challenges is getting appropriate samples for assay development and validation. In particular, cell and gene therapy often targets relatively rare diseases. So if you're developing a bioassay for the clinic where you want to have validation that stands up to regulatory standards, you really need appropriate matrix samples. And often you can source samples from normal subjects. You can use synthetic materials with spiked in target analyte to create contrived samples. But for a lot of regulatory frameworks, validating an assay for the clinic, they really want you to have real matrix samples from patients. And so getting a hold of those samples in the appropriate volumes and at the appropriate time to be able to incorporate those into a development and validation effort can be really challenging. Usually the biorepositories out there and providers of biological samples have a limited ability to do that. And so it really involves reaching out to KOLs and physicians that are interacting with the patients in those communities on a regular basis and working directly with them at maybe a center of excellence for that specific disease area to get access to those samples and incorporate them into the assay development process. I think we may touch on this a little bit later in more depth, but I would also say one of the big challenges in early drug development outside the realm of bioassays is, finding appropriate animal models. As I said in my previous answer, the job of translational medicine is really to connect the data that you generated in your preclinical studies with data in the clinic. And if you have an animal model that doesn't really closely replicate the human disease, it can be very difficult to make predictions about what you're expecting to see in the clinic and how much of a response you're likely to get from a specific dose, which makes phase 1 trials a lot more important and a lot more critical to the efficacy question than they might be in other indications, because it may be your first peek into efficacy in a a disease model that actually really replicates what you're expecting to see in your patients. Your patients are really the only disease model that matters in some instances. And so that phase 1 study may be the first time you're really able to connect the dose to the PK/PD to the biochemical biomarkers and the response that you're seeing in clinical efficacy. And it's very risky to go into the clinic without having that kind of information. But in some instances, there's really no choice.